da Universidade Federal do Rio de Janeiro. E a convite do professor Manuel Luiz Costa, eu estarei apresentando um dos ciclos desta cátedra do Carlos Chagas Filho, que é dedicado às fronteiras da biologia e da medicina. Tá? Hoje nós temos dois ilustres convidados, um deles não fala português, então nós vamos mudar para o inglês, tá? Mas antes disso, o professor Manuel me pediu que fizesse um aviso aos alunos sobre o preenchimento dos questionários que estão no site, verdade? Então, a cada sessão é suposto a cada aluno preencher esse questionário e o ritmo do questionário está bem aquém das participações que nós registramos online, então uma grande percentagem dos alunos não está respondendo aos questionários e nós precisamos dessa informação para validar, né, a presença e a participação neste curso, tá? Então, esta informação fica dada. Qualquer dúvida podem contactar alguém, o professor Manuel seria o ideal, mas a mim mesma também, a gente vai tentar resolver. É um formulário que foi feito, é relativamente simples, e só que tem que ser respondido a cada sessão, tá bom? Isso está um pouquinho aquém do que nós estávamos esperando, tá? I will switch now to English, and before I welcome our speakers, I would like to thank Professor Manuel Luiz Costa for the nice opportunity to participate in this year-round event. And now, of course, I would like to thank our speakers, Dr. Valentino Gantz and Dr. Attilio Pane, for accepting this invitation to be here today and share with us their views and their knowledge on genomic edition with CRISPR and non-coding RNAs. I want to share my screen now, sorry, forgot. Up. Estão vendo, né? Everybody can see this? Yes? Okay. Yes. Thank you. So this is the first session of this wonderful cycle I'll be presenting year-round. So frontiers in biology and medicine. Today, we have the first of many, so stay tuned, okay, because the program is going to be great, and today is not less great. So I'm very happy to present our speakers. We have Dr. Valentino Gantz, who will speak about genomic edition with CRISPR. Dr. Gantz is an associate research scientist at the University of California, San Diego. His lab is interested in gene drive technologies. He uses the fruit fly as a main model, I believe. He is developing genetic strategies to control and reverse RNA-guided gene drives. Dr. Gantz, he graduated and has a master's degree in biotechnology from University of Bologna in Italy. And then he moved to California to do his PhD in biology at UCSD. And then while he was a grad student, Dr. Gantz developed a new method called the mutagenic chain reaction, a CRISPR-Cas9-based technology with an enormous potential in controlling mosquito spread diseases such as malaria and dengue. And then he went to continue his studies as a postdoctoral researcher at the same institution, where he now has his own lab. So I'm very pleased to have you with us today, Dr. Valentino Gantz. And I will present the second speaker of the day, Dr. Attilio Pane, who will speak about non-coding RNAs. Sorry, okay. Dr. Attilio Pane is a faculty member at our own university, so Universidade Federal do Rio de Janeiro, where he also coordinates the Cell and Developmental Biology Research Program. Dr. Attilio Pane works in new generation sequencing and other molecular biology technologies using the fruit fly and other models as a main working model to study biogenesis and functions of non-coding RNAs. Attilio, he graduated and did his PhD in Italy at Università di Napoli. He then went to do his postdoctoral research at Princeton University in the US. And a few years back, he moved to Brazil, where is now a faculty member. So I'm really glad you could join us today. And with no further up, I don't seem to be switching, sorry. So the last slide was just how we're gonna do it today. So we will start with Dr. Valentino Gantz. We will then have a few minutes for discussion. Then we will have Attilio Pane to speak about non-coding RNAs, and then we will have also a discussion. So I'm having trouble with my presentation, and I will stop now because the most interesting part is about to start. So Valentino, if you're ready, please.
Yep. Sure. Okay, can you all see my first slide?
Perfect.
Good. Okay, so today I divided my talk covering like a little bit going into the history of what is CRISPR and what is the potential of this technology, and then I kind of thought to touch on different aspects on how this technology can be applied. One, for example, is in basic research. So how can this technology be useful in the lab for everyone to like look at specific aspects of their own research project. Also, I'll describe how this technology is being considered for tackling some diseases in people and how there are different human therapies that have been developed based on it. Also, like, this is something that's not, it's starting to be a little bit more mainstream, but how this technology can be used to edit crops or animals for generating more productive crops or animals with better characteristics for human consumption. And lastly, I'm going to be touching about a public health application of the CRISPR technology, which is what we're developing at UCSD, which is the use of gene-drive systems to fight mosquito-borne diseases. The example I'm going to be doing is specific on malaria. So I'll jump into the first section of the talk, which is kind of covering what is this CRISPR. Has been popping up in the headlines on a lot of different, on a very wide variety of topics. I mean, people have shown how this could be used for cure blindness, avoid potatoes from oxidizing, modifying all sorts of plants, save the corals, and perhaps even making giant gorillas like Hollywood has to think. So it seems a technology can be doing anything, but what is it in, like, how it came about? So the term CRISPR stands for clustered regular interspaced short palindrome repeats. It's a mouthful, and Cas9 means CRISPR associated protein 9, and these are really the two basic acronyms I'm going to be using. Like the same as in humans, like humans have specific viruses that attack our body. And we also have a adaptive immune system that uses white blood cells that express antibodies that specifically recognize this virus and neutralize them. So also bacteria have phages, which are their version of viruses, that can attack the bacteria and kill them. And so CRISPR is their answer to this attack. So it's an adaptive immune system that bacteria have developed to basically attack phages and then remember, at least the bacteria that survive, remember the attack of these phages, and then are protected from downstream attack from the same type of phages. how the system works in the bacterial cell is, can you see my cursor here? Is it obvious?
Yes.
Okay. Okay, just checking. So how the system works is that once there's a lot of noise.
Só minuto. Alguém pode desligar o microfone, por favor?
Okay. Once a phage attacks a bacteria, it injects its DNA inside a cell, and usually what this infection results in most cases is to the bacteria dying by like an extreme replication of this phage inside it. But in some when a CRISPR system is present, what can happen is that segments of this bacteria, of this phage, get integrated into this specific loci, which are these CRISPR locus, which are these clustered, regularly interspaced palindromic repeats, which is where the name comes from, are these like very repeating elements in which like each fragments of different phage infections are inserted and create these loci. And this is usually associated with a bunch of Cas genes, which are genes that then activate this immune system. Once the piece of DNA is inserted, then it gets transcribed in this pre-CRISPR RNA that generate all these like repeated sequences, and then it's cleaved into small units, each single piece of which the variable part of which targets a specific phage. And so once you have a second round of infection, for example, from the phage green or the yellow one, you do have this CRISPR system that is already programmed to target them. And so at their arrival, the Cas9, which is here in blue, and the CRISPR RNA target and destroy their genome before they manage to reproduce themselves. So every bacteria that survives one infection, it is then protected from downstream infection from the same phage. And similarly to us and other animals that do have different type of antibodies that work a little different, so for example, like dual chain or single chain antibodies in camelids and sharks, there is also like all sorts of different Cas systems that have developed in different types of bacteria. There are very complex ones that require several of these Cas protein to assemble and bind this guide RNA that induces the cut, or there are more simple ones like the type 2A and B that do have this one big protein, Cas9, which does pretty much all the functions of finding the target DNA to destroy and cutting it. And this is what has been then used downstream as a tool for genome engineering. Now let's go into more the application. So how this bacterial adaptive immune system is used in real life. So the three basic components that in research we're keeping in mind is the Cas9 nuclease, that is the enzyme that performs the cut. Second, the guide RNA, which is that small RNA that in the bacteria recognizes the phage, but in this case, it can be designed ad hoc to target any wanted genomic target. And these two components can combine, and then the Cas9 itself uses the guide RNA to find the location of the target, and then has a nuclease activity in the protein that induces a cut at the precise location. And this interaction results in a double-strand break. Now, once this double-strand break has happened into a cell, the cell, it's not able to survive with a broken chromosome, let's say. And so there are multiple different DNA repair systems that can act at this point. And the main two that people have been taken advantage of are the NHEJ or MMEJ. It's not clear how these two, which one of these two systems is prevalent in certain situations, but each one of them usually results in a small mutation. So wherever you're using the CRISPR-Cas system to target the genome, it does disrupt. Let's say if it's in a specific genes that you're targeting, it creates a small mutation that invalidates the gene. Or separately, it's possible to use homology-directed repair that allows to insert something within the cut. But in normal situation, what HDR uses the intact chromosome that doesn't have a cut or a mutation, and uses that sequence to gap the broken chromosome, and therefore copies accurately the sequence from the companion chromosome. And this system has been so impactful because compared to previous technologies that require a lot of engineering of different molecules, such as zinc fingers or endonuclease, in this case, the Cas9 enzyme is very big, but it's always the same. And what changes in the system is this small RNA that can be easily synthesized, and that acts kind of like as a key. So uses the sequence that is put on this guide RNA guides the Cas9 to target any potential place in the genome. And so it is like feature of CRISPR that has been making it so impactful and widespread as a technology. And the second thing that is also worth mentioning is the fact that it has been tested in pretty much any organism and has worked perfectly. So there is, I currently am not aware of any organism in which this system has not worked. So it's something that makes it also extremely appealing. And this is also reflected by the amount of papers that have used CRISPR, and actually this graph arrives to mid-2014, so it'll be interesting where that is right now. But the two previous technologies that we were using to target genomes, like talons and zinc fingers, have slowly dwindling, and the CRISPR publication really shot up. And similarly, also the amount of funding for CRISPR project has shot up accordingly, as well as patents that have been published for this technology. But while all this, big hype of this technology started after the publications in 2013, showing how this technology can be used as an easy tool for generating gene edits, the history of CRISPR is way older, with the first report of this weird genomic loci in bacteria that had this like repeat pattern in 1987. And then later on has been recognized as a system that caused adaptive immunity in bacteria. This was actually done in 2007 in a yogurt company in France. So it was a very wide story. Then it has, in 2008, it was shown that CRISPR acts upon specific DNA targets. And then later it was done, it was like characterized like exactly like what these targets are composed of. Specifically, there is this PAM component that is very important in how the Cas9 system cuts. And then later on, how the system was engineered and ported from a bacterial system to becoming a tool that can be used to edit eukaryotic cells. And in these publications, they showed like how this can be used to edit human cells. And so it was really from that publication that then all sorts of downstream applications, such as they use for basic research and for human therapy, came from. So I'll I'll now talk about like a couple of examples on how this can be used in basic research, and then I'll jump into the other applications. Like this is one of the things that I remember really was blowing my mind about one of the applications, the first applications of CRISPR. And this was a paper in which they've identified a specific regulatory sequence of the sonic hedgehog gene and how this has evolved over time in different animals. And it has basically degraded into snakes. And what these authors decided to do is basically target a specific regulatory region in the mouse and see whether removing the function of it would result in a phenotype like cobra and python. And they've shown that, while in regular mouse you have the development of limbs, just the small, very, very small alteration of one particular DNA sequence that alter the expression of this gene results in no limbs developing in mice. And so this, with a very simple experiment, it was shown like how a regulatory sequence of a gene was able to slowly transform an animal that had 4 limbs to one that didn't have any. A separate application of CRISPR that instead is more powerful, and it has been later developed as part of like further applications of this technology in the lab. And it's this concept of a multiplex approach to screen specific phenotypes in cells. And this has been done in insect cells and human cells. But the basic idea is that there are, in the first step, there are a bunch of guide RNAs that are designed, and then they're cloned into specific cells in a 1-to-1 ratio, so that each specific transformant would have one guide RNA only that targets a specific sequence, and this sequence can be known because if you sequence the specific cell, you will know what guide RNA that cell had. So once these libraries are then created with CRISPR sequences that target specific genes, they're then put into single variants, and then they're transfected into cells. Then what you have is a big library in which each cell expresses specific guide RNA. And what you can do, you can submit this library of cells to any sort of perturbation. It could be heat, it could be infection by a disease or action of a specific drug. And after the exposure to your experimental condition, you can take all the cells that survived and deep sequence to see what guide RNA are remaining. And by comparing what guide RNA are remaining in the population, from which the ones that you started, you can basically go back and figure out which genes or which genomic locations those guide RNAs were targeting, and then you can look and identify those potential candidates for the effect that your experimental approach has then generated. Now I'll go into the human therapy, in which here people have instead specifically targeted known mutations. So there are several diseases that are caused by small DNA alterations that are known and very well characterized. And from the normal DNA sequence, you have a specific point mutation that generated a specific phenotype. And in this case, it's the example of sickle cell anemia. There's a lot of different diseases that are caused by either point mutations or small aberration in genes that can also cause like dominant phenotypes. What people have tried to do is target specific location and then try to revert this phenotype from the disease back to the normal phenotype, and have done so by delivering these CRISPR components in humans. And it has been done in two slightly different ways depending on the disease that has been targeted. There is direct delivery, which is called in vivo editing, and basically the CRISPR-Cas9 components are delivered with viruses, and they will target either systemically or a specific, or they're like programmed to be delivered to a specific tissue that needs to be edited. And this system delivers the CRISPR-Cas9 components in that specific tissue that needs to be edited. The second option is instead to extract cells from the patient, modify them with CRISPR-Cas9 in the lab, and then once you have selected clones that have the wanted modification, you can put them back into the body where they will divide and colonize. So, for example, in the case of, yeah, I don't want to go too much into detail because I think I'm already going too slow. But each single one of these does have a specific consideration that you have to do. For example, in the case of an ex vivo approach, you do have that you remove some cells, that you put them back in the patient, and so there is no there's a little bit less risk because you know exactly what you're putting back into a patient. While in an in vivo system, you're delivering viruses like intravenously, and you really might end up having some effects that you're not predicting. The other consideration that you need to consider is that whatever modification you're doing, for example, in an ex vivo approach, you're going to add some somatic cells that will be present in the person, and they will not be passed on to the offspring. While instead, if you're delivering a virus into an entire patient, you have the chance of modifying germ cells and therefore delivering whatever mutation you're trying to generate also to the progeny or to the kids and daughters of that person. And currently, since there is very much unknown risk, the editing of germ cells is something that no one has been pursuing, except a couple of publications from a controversial publication from a group in China that has edited the 2 human embryos, which made a lot of noise a couple of years back. I also wanted to touch onto some sort of second generation CRISPR system. In this specific case, the Cas9 has been modified to be a dead Cas9. So the nuclease activity has been completely abolished, but this molecule is still capable of using the guide RNA to find a target that is designed to recognize, but it's not capable of cutting. What these authors have done is that they have attached to the Cas9 a cytidine deaminase. And what this does is, upon recognition of the genomic target, the cytidine in this strand is then converted to a uracil, and then the cell, upon DNA replication, turns that uracil into a T, and therefore it has a chance of converting a C to a T after the specific editing. And people are looking into the use of this specific tool for editing single base mutations that create a specific disease, such as the one for the sickle cell anemia. And if you compare this base editing system to the regular CRISPR, when you do target with CRISPR, you end up generating all sorts of this changes in bases or small insertion, deletion, usually deletion close to the cut site, which is about here. And you can see that when you instead use the same exact targets, but you use a base editor, you have almost none. There's only one case here in which you have a small deletion, but you do have a very targeted modification of what your action is meant to do. And so this is another second generation CRISPR DNA editors that people are considering for the use in therapy, because it is way more precise than regular CRISPR. I also want to touch on the editing of crops and animals, as while there is a lot of like hoops that we need to go through for human therapy, and it will be very hard to get to a specific application, but the use in crops and perhaps in animals might come sooner to impact all of us. And one of the things that people are considering is just engineering specific traits into cows that would allow us to maintain cows in like bigger herds, and also making them more productive for what concerns both the meat production or milk or whatever we would be interested. One of the things that people have been exploring is the generation of cows with no horns. Like currently, the horns of cows need to be cut, otherwise when they're in big herds, they can injure themselves with the horns. And so by not having horns, people that own big herds, they don't really have to chop off the horns every, 6 months or whatever they would have to. Also, you could engineer all sorts of animals to have more muscle, so they develop more muscle, they create more meat, so you have way more meat generated per animal. This are specific bulls and pigs that have more meat than their wild type counterparts. Also, people have been exploring the generation of better fats like omega-3, which are in very low amounts in the milk of cows, but it could be integrated just by supplementing additional enzymes, or less lactose for people that are lactose intolerant, or more milk protein so that it becomes more nutritious per unit. Additionally, like this is something I also have seen, like human lysozyme is an enzyme that can be used, but it could be easily produced in milk and then purified. So there are a lot of things that could be engineered into the animals for our benefit. And similarly, in specific crops, there's been a lot of gene editing in these animals, but like with the advent of CRISPR, a lot of mutations have been easily reproduced, such as the increase in production just by adding more branching in tomatoes, for example, or more amount. So like by increasing the yield. This is something I mentioned before, is the no browning. I personally don't care, but some people do. When their apple goes brown, or their potatoes, or same as for the mushrooms. By removing specific genes, you do have that this fruits and vegetables don't brown anymore, and they just look better in the store, I guess. Other things such as the rice with added beta carotene. This was actually done previously from CRISPR, but I put it here as an example of what could be done in also grains, just adding vitamins that are non-present to make specific foods more nutritious, as well as other things like pathogen resistance, which have also done in the era before CRISPR, but now can be engineered specifically within the plant way, in a way easier way than has been done previously. Now I'll go into the last section of my talk, which I'm going to be going over what we are working on at USCSD, which is the use of this gene drive technology to fight mosquito-borne diseases. The basic construction of a gene drive uses these two components that I mentioned before, the Cas9, which is a nuclease, and the guide RNA, which is this addressing molecule that tells the Cas9 where to cut. And we've built a construct, a synthetic construct here below that has these two genes placed in between two stretches of DNA that are homologous to whatever sequence is targeting, this gene drive is targeting on the genome. So that the Cas9 and guide RNA combine, cut at that precise location that the guide RNA tells the Cas9 to cut. And now we use that HDR, or homology-directed repair, which is that accurate repair system that the cell uses to fix that gap. And instead of using the other chromosome to fix the gap, it uses this synthetic piece of DNA that we have added. Now, though once you have this gene drive construct inserted, you now have that this property keeps going. And so like once it's inserted in one chromosome, the construct by the same exact mechanism is capable of cutting and copying itself onto the other chromosome. And if now this animal that has two copies mates with a wild type animal that has only one, the same process will keep going. And so if you compare a gene drive inheritance to a Mendelian inheritance, you have that if an animal carries this element, it will pass it on, because it has only one element, it will pass it on to 50% of their progeny, and generation after the other, it maintains the same frequency in a population. While instead, if you have this property of copying itself onto the other chromosome, now it's not passed on to 50%, but to 100% of their offspring. And then it copies again. And so slowly, this sort of gene drive element can spread into a population. And since they double their frequency at each generation, you have that they follow a logistical curve with an exponential growth of their frequency in the population. And we've modeled this for mosquito population. And as you can see, is that if you release only one, I think was 1 in 1,000 animals in a population, you have that you can quickly go from 1% to 100%, close to 100% in as little as 10 generations. And this sort of like spread of a genetic component was a sort of a holy grail that the mosquito field was looking for. And this is because these gorgeous animals are, ferocious, blood-sucking, but also can spread all sorts of nasty diseases. And they're caused every year to about 830,000 deaths, which is way more than even people cause. So they're really the #1 killer animal in the world. And out of this deaths, the biggest majority is caused by malaria, which is an intracellular parasite that is capable of attacking and chewing up the red blood cells that we all have. It is carried by mosquitoes, but not all mosquitoes can carry. Only the genus Anopheles carries the Plasmodium. It is very specific to humans, so this means that it's not going to be carried by other animals, only by people, and it cannot survive in the wild. So it is an obligatory parasite, so it either survives in the mosquito or in the person. And this sort of cycle has given us an advantage to generate drugs to kill it within the person, or pesticides to kill the mosquitoes that carry it. But these mosquitoes are not born with malaria, so once they're hatched as freshly new mosquitoes, they need to pick up the disease into an infected person and then vector it to a second person. So that's how the disease is spread. And it's still endemic in several countries, most of which are in the tropical regions. There have been over 200 million cases with about 500,000 deaths in 2015. And this disease currently creates the biggest problem in sub-Saharan Africa, where more than 90% of the deaths occur, and most of which are children under the age of 5. And while we have developed all sorts of different technologies, such as bed nets with pesticides on it, there's fumigation happening, and there's drug to both kill the parasite or cure it after you have contracted the disease. These methods are slowly evolving resistance, either in the mosquito or in the parasite. And it has recently resulted in a small uptrend of the yearly cases. So until like 2013-14, the cases were steadily dropping, but in the recent years, the trend has been slowly reversing. So this gene drive technology is something that we could use. We could add it as a tool to the arsenal to fight these diseases. So to go a little bit more in detail on how this works, I already mentioned this before, but the idea is that when you have a specific trait, such as this blue gene, and this blue mosquito mates with a wild type one, you have that 50% of their progeny inherit that chromosome, same as would be for people. And so generation after generation, this trait remains in the population, but remains at a very small frequency. So if you have a beneficial trait, it will not have a big impact into an entire population. And this follows the same Mendelian rules that were described by Mendel a long time ago with peas. Well, a gene drive defies these rules. So in this case, we have a gene drive with this, let's call it a green gene, that is capable of cutting and converting the other chromosome. So now you don't have one, but you have two of such chromosomes in this individual, and they're given off to 100% of their offspring. And since you have this conversion again, you have this amplification of the gene drive at each generation, leading to slowly the conversion of an entire population to having this new green gene. And what we have done is we've generated mosquitoes that do have a gene drive, that do have exactly this property. And on top of it, they have this green gene, which is a genetic vaccine for the mosquito. So the mosquitoes that we have built can bite an infected person, but they cannot harbor the plasmodium. So they can be around, they can be healthy, but they cannot transmit the plasmodium anymore. And this was done in collaboration with Anthony James at UC Irvine. And what Tony has done is build this specific mosquito that have a few modifications so that when the plasmodium gets sucked in with the blood meal of these mosquitoes, the mosquitoes express in the lining of their intestine this antibody that binds them and prevents them from moving into the lymphatic system where they need to go back to the salivary glands. And even if one of these guys manages to escape and enters into the bloodstream of the mosquito, there is another antibody there ready, attached to a toxin, ready to kill them before they make all their way to the salivary glands. And we have shown that this gene, so it is an engineered gene that has these two antibodies that are expressed in mosquitoes attached to a gene drive, is capable of spreading into mosquito population in the laboratory. And you can imagine that very few mosquitoes like these could be released into the environment, and slowly you can convert an entire mosquito population to have this novel trait, and pretty much having all mosquitoes in a given region that cannot spread a disease anymore. And this sort of strategy was engineered for Anopheles, which is a mosquito that does have a very advanced genome editing, genome engineering knowledge, but the same kind of concept could be used for all sorts of other mosquito species that spread a lot of other diseases. And given the current global warming situation we have, that the mosquito problem is not going away anytime soon. So we can use all sorts of pesticides we want, but it's not going to go away for good. They're going to be here to stay. And so we just need to figure out ways of dealing with them, and perhaps just targeting their capability of carrying the disease rather than actually removing the insects. And this is something that I believe is what we have been doing so far, is that we have our target mosquito, and we're using pesticides that don't affect that mosquito only, but affect all sorts of weird bugs in the environment, especially other animals that might be beneficial, such as bees. And not only they affect those animals, but also animals and plants that feed or depend on those insects. While a technology like a gene drive is something that is designed specifically to target the animal that gives a problem, and therefore it's a much greener solution to all what we've been doing so far, which is dumping a bunch of pesticides in the environment. And with that, I'm gonna end my presentation, and I guess we can go to the discussion. This is an acknowledgement slide for all the people that are working on these sorts of technology here at UCSD. Okay.
All right. Thank you so much, Valentino. It was a very good talk. I'm opening the session for questions. So, se alguém não está à vontade para falar em inglês, pode perguntar no chat ou em português que eu traduzo, tá bom? Não perca a oportunidade de perguntar alguma pergunta. Tá valendo qualquer língua. Italiano ele também deve entender. Então, anything, right? I'm just saying, if people are shy to ask in English, they just can go ahead and ask in Portuguese. So.
May I?
Please, Elena.
Okay, so I always enjoy your talks, Valentino. I have a general question for you, which is in relation to how do you feel that the CRISPR technology, not the CRISPR technology, more specifically gene drive technology, is being accepted or not by the general public? And I'm asking that because it involves not only the editing, but also placing genes that are not from the animal itself, and thus you're using gene editing you're using CRISPR editing for putting inside transgenes. So do you think that's simply by not involving the word transgenesis, let's say, there's a difference in the public acceptance or not, regardless of the question about the gene drive being able to copy itself? And also you can think about the same thing for CRISPR if when you put something inside the animal, biomolecules recombination, that was not there.
Yeah, well, in this specific example that I made is on top of the gene drive, you're using this new engineered trait. But if you think about it, it's like any of this CRISPR-based gene drive is inherently on its own a transgene already. So it's kind of like if you do use this technology, you're already locked into generating transgenic animals. And if used as a gene drive, it has the potential of spreading into an entire population, and it will become the new norm. There are technologies that have been developed by other groups that instead use a gene drive to spread a deleterious trait. And the idea there is to modify, or I guess you're suppressing a population by spreading this specific negative trait. And at the end of the spread, since this trait is deleterious, it will slowly go away. And what you're left with is a dramatically reduced in size population. The drawback of that approach is that while you don't have a transgenic or genetically modified population at the end of the process, but you do have wild type animals, very few, but those wild type animals will reproduce, and in a couple of months after, you're back where you began. Mm-hmm. So it's something that could work in a localized area. For example, on an island, if you have malaria, you could wipe out all the mosquitoes, and then if you have 2 or 3 left that were not carrying malaria, then you still can eradicate the disease, but not eradicating the mosquitoes.
Valentino, 2 questions popped up in the chat. I will read them for you. So Mateus asks, besides the structural differences between the CRISPR systems reported, what are the functional differences between them? Like in the systems with more elements, are there less off targets?
This is something that I don't think have a clear answer. There has been, first because I'm ignorant on that point, except of maybe one other system. But there have been other systems described, such as the one based on CPF1, that does have a different cutting pattern. So while Cas9 does generate a blunt end cut in the DNA, CPF1 generates a staggered cut, which could be used for a better HDR efficiency, or could have a different pattern in repair. There are the original CRISPR system that have been shown in other bacterial species that do have multiple components, and has been shown in some of these that like the guide RNA that these systems use is much longer. So it's about, I think, 32 to 33 nucleotide versus the 20 that the Cas9 uses. And so potentially you do have that some other systems that have a longer guide RNA might have less off targets, because they have this bigger requirement of sequence identity or matching with the target.
We have another question from Ana Celia Castro. I will read it to you. Great talk. My question relates with gene therapy. Do you have news on CRISPR being applied to rare diseases, correcting the wrong gene, the defective gene? And she has a second question. The applications in agriculture are amazing. Do you know about enterprises using CRISPR in the meat supply chain?
Okay, I'll get the second question. I'm not aware of any specific company. I know that there are groups that are exploring the use of CRISPR in the meat supply chain, but I don't know of any company that kind of came out with a product, and or maybe I missed it, but I'm not aware of any specific one. While there is several companies that have taken CRISPR and applied to specific rare diseases. There is Editas Therapeutics, for example, have a is applying in vivo editing to a rare disease that causes blindness. So what they're literally trying to do is injecting CRISPR components built into a virus directly into the retina, and with this hoping to cure this disease. There are also other applications that are targeting dystrophin mutation causing muscular dystrophy, and there I believe there's several groups that are looking into this, and depending on what mutation you have on dystrophin, it could be edited in different ways, but it's a little bit of a more forgiving kind, because in some situation you have a point mutation that creates a stop codon in an exon, but that can be just chopped off. If you chop off the entire exon with CRISPR, which you can just cut on each side, you are missing a part of the protein, but missing a small part of that protein, it's much better than having the rest completely abolished. So by removing an exon that has a stop codon, you're recreating, you're reconstituting the frame, and therefore like those edited cells are then much more functional than one that had a point mutation.
We have time for one last question. Ina Pacheco asks, the target sequences used in CRISPR are very small. How to deal with off targets?
There have been different discussion on these off targets, and it's something that is more relevant in some application than others. For example, I don't know, I'm working with Drosophila. When we're designing a guide RNA, we take more care about like if it does cut where we want, rather than if it does have off targets. There's always a chance of off targets, but in a lot of cases it's not terrible. It's good to be aware of them. In situations like editing of crops, you might also not care because you can just select plants that don't have off-target effects. But when you're doing something like a human therapy, you do want to really screen for off-target effects. And there's been a lot of characterization of off-targets given by specific guide RNAs, and there is software that you can use to like design guide RNAs that don't have off targets or have very little off targets. But there is always a small chance of, delivering a very strong nuclease into a cell. You always could have also off targets. There is also reports of the Cas9 on its own having a mild nuclease effect without the use of any guide RNA. So there is all sorts of issues also with that. Even that base editor system that I showed, even if the Cas9 is completely dead, the addition of this deaminase attached to the Cas9 induces a background mutation that's much higher than regular conditions. So while it's very promising for the use in therapy because it has this very precise editing, it does increase background mutations. So people are now reconsidering also that.
Okay, we keep on having questions, so I will allow a last one. Okay, so we go on schedule. Alain, he asks, can the CRISPR multiplex approach be applied to cells in vivo or only to cells in culture, or maybe in ex vivo slices at least?
I don't know. I've seen it that I believe it could I've seen it done only in vitro with like cell pools, but I'm sure someone could come up with an approach that could be done in vivo. Definitely on animals that are very fastly reproducing, they could imagine to have like fruit flies with different guide RNAs and then screen them. But most likely it's easier to do with even smaller animals like worms or bacteria. So.
Okay, Valentino, thank you so much. We will now move on to our second speaker of the day, Attilio. Are you there?
Yes, can you hear me?
Ready? Okay.
Can you hear my screen?
Attilio.
My screen?
I can see your screen. You have to just put it on the presentation. Looking good.
Can you see it now?
Yes. You have to speak louder, maybe.
Okay, we have the same problem as earlier on. Okay, then thank you, Caterina, for the introduction, and I want to thank also Manuel and the organizer for inviting me to be part of this seminar series and the activities of this group. It's really an honor to be here. So my intention today is to talk about other types of non-coding RNAs. We have seen with Valentino how a specific mechanism that was discovered in bacteria was then harnessed to actually attempt to cure human diseases and also for many other applications. Now, there is many other different types of non-coding RNAs that are being investigated and that exert important roles in plant and animal development and often are connected to human diseases. So my intention is to show you these other, a few other non-coding RNAs, but also I want to emphasize the role of different model organisms in dissecting the molecular mechanisms and the function of regulatory pathways and signaling mechanisms in general. So I will start with let's see if the presentation works. Okay. So I will start with this famous dogma of molecular biology that was proposed by Francis Crick. And the dogma states that the genetic information that is contained in the genomes flows from the DNA to the RNA and then from the RNA to the proteins. And this was the 50s, was the 1950s. But a few decades later, the discoveries from Sidney Altman and Thomas Cech, who identified RNAs with catalytic activities, challenged that dogma, and they brought about a new perspective on what a gene should be considered. So they received the Nobel Prize for this discovery, for discovery of the ribozymes, which changed the view of also what of life could have appeared on Earth. Now you had an RNA that had both the ability of transferring genetic information or carrying the genetic information, but at the same time, the RNA had a catalytic activity, so it could work as an enzyme without being a protein. Now, this was extremely important because in the genomes of both plants and animals, you have a lot of genes that are transcribed, and you have intergenic regions that were considered junk DNA, which produce transcripts. These transcripts do not encode for proteins, but still, they have a function. So it's this field that has attracted a lot of interest over the past decades. And I will show you a few examples. Now, the fact that Valentino talked first helps me a lot to go faster, to go quicker through some of the slides, where I meant to show you the basic principles of how a non-coding RNA generally function. So you generally find a non-coding RNA in a complex with the proteins, and the RNA is the guide. It's the eyes of the protein complex that will then perform an enzymatic activity. The RNA, the non-coding RNA, can share complete or partial base pairing with the target RNA, and it generally leads to the silencing of the gene by either cleaving the transcripts, by silencing the translation of the RNA, or by introducing chromatin modifications. So these are just general rules. And how about the mechanisms also I've already mentioned? What about the functions? So there is really a range of functions that non-coding RNAs exert in the development of the organisms. And these functions range from gene regulation to the maintenance of genomic stability. And I'll show you some of the pathways that control these aspects of the development. But also they are involved in chromatin assembly, antiviral defense. And again, Valentino told you about some mosquitoes that are vectors of human diseases like the Aedes, that is a vector of Zika and dengue. So non-coding RNAs act in this species as antiviral mechanisms. And then in worms, we have the recognition of what is self from non-self, and then many other roles, many of which are still being investigated. Now, this is a schematic of how many different types of non-coding RNAs we have in the living organisms. And Valentino talked about the CRISPR, and I will tell you about mostly some of the RNA silencing mechanisms that involve microRNAs, piRNAs, and siRNAs, or like viral siRNAs, as well as a couple of long non-coding RNAs. Now, when does this field really take off? Most of the milestone discoveries were done in the 90s, or at least some of the most important discoveries were done in the 90s. For example, the first microRNA to be identified, LIN4, was cloned by the Victor Ambros group at Harvard. And this microRNA was specific of worms to the point that at the beginning, it was thought that microRNAs were specific of this species. It was not a conserved element in nature. But then a second microRNA called LECT7 was identified again in C. elegans. And now LECT7 is very conserved in the evolution. You can find the ortholog in human, and it is associated with some diseases. I will show you one later on. Now, these mutants, these RNAs, these microRNAs were identified because mutations that impair their function generate the so-called heterochronic mutants. When the worm does not have a functional LIN4 or a functional LECT7, the development is altered to the point that the transition between one developmental stage to the next is either faster or slower. So how about the biogenesis of the microRNAs? So after the discovery of the first microRNAs, the effort of many labs led to understand more of how they are produced. And in this slide, I've already listed the other mechanism that I will talk about today. And you can see that they share some common components. The dicer proteins that are ribonuclease enzymes that are involved in the biogenesis of microRNAs and siRNAs, and the argonate proteins that basically take part to the biogenesis of the piRNAs and the downstream silencing mechanisms in all of the branches of these RNAi phenomena. Now, for the microRNAs, the substrates that leads to the production of the mature molecule is a stem and loop structure. MicroRNAs are genes, so they are transcribed from the pol 2, RNA pol 2, but the RNA folds back to form a stem and loop structure, which is then processed to produce the 22 nucleotide mature microRNA. And now the microRNA is incorporated into a complex called either microRNP or MyRISC that is centered on the argonate 1 protein. And now the complex will recognize the target RNA and drive both the translational inhibition and the RNA degradation. I should mention that this slide is referred to the pathways that act in the fruit fly, Drosophila melanogaster, where the three branches are quite separated, although there is a certain degree of interplay. In other organisms, like in human, for example, there is only one dicer enzymes and 4 argonates, while in the worm, C. elegans, you have 27 argonate genes. So there is a lot of variability in terms of number of argonate genes in distantly related species. All right. So I told you about microRNA in worms. What happens in human? This work from Dave Bartel lab showed that approximately 60% of the human protein coding genes is potentially targeted by microRNAs. So this really emphasizes the importance of microRNAs in human development. Many times there is redundancy. So the same microRNA can target different genes, and conversely, the same gene can be targeted by multiple microRNAs. We can talk about, again, the molecular mechanisms in more detail in the question session. I want to tell you about a specific microRNA, so to give you really more details about one specific mechanism and how it can be used in the medical field. There is approximately 5,000 microRNAs in a human, many of them are predicted, and a study where these microRNAs were investigated in 64 tissues and organs allowed to place them in different beans, like housekeeping microRNAs, which are expressed in all tissues, and then microRNAs that are expressed only in a few tissues. And then there is another bean which contains microRNAs that are specific to a certain organ or tissue. So one of them is MIR182, which is specific of the liver. And a lot of studies have shown that microRNA coordinates a network of regulatory events, which is important for hepatocyte differentiation, cholesterol metabolism. MicroRNA MIR122 protects liver from hepatocellular carcinoma, but is also involved in viral replication. You can also see from this slide that there is a lot of feedback loops, feed forward or negative feedback loops. And for those that followed the seminar from Professor Bruce Alperts a couple of weeks ago, you remember that he emphasized a lot the importance of these feedback mechanisms in the evolution of gene regulation. Now, what happens if we remove MIR122 in a mutant mouse? Now, the mouse is actually fine, but it is more prone to develop a number of diseases, like alcoholic liver disease, again, hepatocellular carcinoma, and others. So the microRNA has an important role in protecting the liver, in protecting the organism from different diseases. What is remarkable, however, is that this same microRNA is used by the hepatitis C virus to replicate in the liver. This is not unusual. It actually happens very often that viruses develop a mechanism to circumvent the antiviral defenses of the host. So in this case, for example, the hepatitis C virus uses the microRNA to protect its 5' region from exonucleases. So XRN2 is a typical exoribonuclease that is found in human cells, but it's very conserved in evolution. What this exonuclease would normally do is to recognize the 5' region of the viral genome and start to chop it. So it will basically digest the viral genome and prevent the virus from replicating in the liver. However, since the liver expresses high levels of MIR122, this microRNA can bind the 5' region of the viral genome and protects the viral genome from the activity of the exonucleases. So basically, this microRNA is not so good in terms of viral replication because it helps the virus to survive and thrive in the liver. So what happens is that these microRNAs can be used, or people thought of using this characteristic of MIR122 to develop a drug. And the drug is called miravirsen. It's in stage 2 clinical trials. The miravirsen is a modified oligonucleotide, antisense nucleotide. You can see what this means in this slide here. This, in the upper part, is the microRNA, and the oligonucleotide is complementary. It's just a stretch, it's a sequence that is complementary to the microRNA. So the idea is, if we introduce this microRNA, this AMO in the organism, it should be absorbed by the liver, and in the hepatocytes, it should bind to the microRNA using Watson and Crick base pairing, and in this way it would sequester the microRNA from the cytoplasm. Now, if the microRNA is no longer available, clearly it cannot protect the viral genome that is now susceptible to degradation by the cytoplasmic exonucleases. And this seems to work quite well. There's been a number of studies that have been done in mouse and primate models where the viral load has been significantly reduced, in some cases by 50%, although just for a month or two. And there is no negative effects, because you can imagine that if the microRNA is at the heart of these regulatory networks, and if you remove it, you have all these possible diseases. then also the miravirsen, by blocking microRNA, could make the organism more prone to develop other diseases like the hepatocellular carcinoma. But this does not seem to be the case, and there has not been undesired side effects from the use of these drugs. Now, talking about microRNAs, after the first findings by the group of Carlo Croce, the microRNAs have been firmly connected to various types of human cancer. And in many cases, like, they are downregulated or they can be upregulated. For example, I told you about lead-7 that was first discovered in C. elegans. You can see that it is downregulated in lung, breast, gastric, ovary, and other types of cancers. In many of these instances, however, we are talking about correlation. So there is either an increase or a decrease of a certain microRNA in a specific type of tumor, but it is not known whether this is the cause of or the effect of the tumorigenesis. And this is, of course, one of the challenges of the future. Nevertheless, the fact that you have different microRNAs that are specifically deregulated in a specific type of cancers opens a new possibility. The possibility being that we can use probably the microRNAs as markers, as biomarkers, to follow the process of tumorigenesis, to characterize a specific type of tumor, but also to follow the progression, either the growth and the metastasis of the cancer, as well as the recovery after the treatment. Okay? So now let's move to the second branch of the RNAi phenomenon. And this is the canonical RNAi interference, RNA interference. Again, we have a dicer enzyme that is involved in the biogenesis. The substrate here is a double-strand RNA molecule. So it sends an antisense RNAs, which are chopped by dicer 2, and this is a processive enzymatic activity which will chop the substrate in different siRNAs. Differently, a microRNA gene generally gives rise only to one microRNA. And you can see that the mature siRNA also is incorporated into a complex that is generally centered on an Argonaute protein. In Drosophila, this is a different Argonaute protein. And the ribonucleoprotein complex called the siRISC that is produced will now use the siRNAs as guides to identify the target RNA and degrade it. In addition to this, the siRNA complex can drive chromatin modification. I should state also, I should underscore that the RNA degradation mechanism that is triggered by this pathway is different from the one triggered by microRNAs. And again, we can talk about it later on. Now, when did the story of the siRNAs and the RNA interference start? Again, we go back in the 90s, and there were some observations that were quite puzzling for the investigators. In particular, there were two studies, one that was performed in petunias and another one in Neurospora, that is a fungi, where it was shown that by introducing an extra copy of the gene in the plant or in the fungi, instead of increasing the expression or the amount of product of this gene, you would actually have a phenomenon of co-suppression, whereby both the endogenous gene and the extra copy would be silenced. So it's basically the opposite effect that you were hoping for. It was called co-suppression in plants and quelling in Neurospora. And for many years, this remained really a mystery. Not for many years, actually. For a few years, this remained a mystery. Here you can see another phenotype that is generated when you insert an extra copy of this gene in the plant. This is a chalcone synthase encoding gene, which controls the color of the flower. Now, you would imagine that if you had more copies of the gene, you would have a darker color. But in fact, in this case, was starting with already a dark flower. If you insert a transgene in the flower, now you have a suppression both of the endogenous and the transgene, which leads to these white patches. In some cases, the flower can be completely white. And yet it shows the expression of the gene that in the presence of the transgenic construct is completely suppressed. Now, We get to 1998, and this was really a turning point for the RNAi field, for the many different non-coding RNAs actually that were later on discovered. Why? The study from Craig Mello and Andrew Fire that received the Nobel Prize for this discovery showed that by injecting double-strand RNA molecules in the C. elegans, so again, the worm was an important model system to understand conserved molecular mechanisms. So injecting double-strand RNA in the worm with a sequence specific to a certain gene of interest, you would end up silencing the target gene very potently. And this is shown here in the experiment from their paper. At the time, it was pretty common to use antisense molecules to try and downregulate gene expression. It's a little bit the same principle as for the antisense oligos, like the miravirsen. So the idea is that if you introduce an antisense molecules with respect to your cognate RNA, so your target RNA, they would base pair, and this structure will prevent the target RNA for being translated. Now, what happened is that accidentally they discovered that by injecting a mixture of sense and antisense RNAs, this would basically base pair, form a double-strand RNA molecule, and now the double-strand RNA would have a very strong effect in silencing your target gene. In this experiment, you can see a worm that is expressing the GFP, so it's green fluorescent. And here is the worm where the double-strand RNA for GFP has been introduced. You can see that except for a few regions, the expression of the GFP is completely silenced. Another important observation that they made is that the RNAi, so the silencing, is heritable. And actually, it can persist for a few generations, even in the absence of additional double-strand RNA sources. So the mechanism can be transgenerationally inherited. And now we go to 99, it was just 1 year later. And this is really another milestone discovery in the field of RNAi. David Volcom was studying plants, and in particular, he used both the transgenic system that I've showed you before, so the system with co-suppression, but also plants that were infected with the tobacco mosaic virus. So plants that are infected with virus or plants that have a transgene can produce short non-coding RNAs that are 25 nucleotide long in plants. And these are the actual effectors of the RNAi. So these are the guys that are silencing the genes, for example, here in the worm, even though in this organism the size is different, but these are the actual effectors. So this is what this was really, really important. And in fact, he showed that in the case of the transgenic line in the petunia, the insertion of the transgene in the genome led to produce short non-coding RNAs that were responsible for silencing both the transgene and the endogenous gene, thus resulting in the patched phenotype. Now, since we have talked about the fact that the RNAi can be a very important tool to suppress gene expression, just want to show you a couple of applications, but also endogenous mechanisms. So the truth is that there is not many siRNAs that are physiologically produced in any organism. And so far, what is really the endogenous function of this pathway in regulating gene expression or chromatin assembly, it's only clear for a few species, in particular unicellular organisms. But what is clear is that the RNAi is a powerful antiviral system in arthropods. So in this case, for example, I'm showing you an experiment that was done with Anopheles gambia, infected with an engineered version of the onion virus. I'm not even sure I'm pronouncing it correctly. But this is the mosquito that is infected with the virus, with modified virus. You can see the fluorescence. And here is the viral load measured by the fluorescence, indicated by the fluorescence, after injecting double-strand RNA molecules against the virus. So you can downregulate the virus by injecting double-strand RNA molecules. Now, there's several labs, and Valentino knows certainly a lot better about this, which are trying to generate transgenic lines which express double-strand RNA molecules, for example, in Aedes against the Zika, dengue, and other viruses, because clearly it could help to clear the mosquito from the virus and therefore reduce the spread of the disease. Now, I'm showing you also some of our data, and surprisingly, we have found viruses in this insect here, which is the vector of the Chagas disease. We found basically 8 viruses recently in the ovary. So this is the ovary of the Rhodnius. We were doing transcriptomic analysis in this tissue, and we ended up by discovering 8 new viruses that have not been characterized. Actually, these are the first viruses to be identified in Rhodnius. I'm showing you here what we call the RPV1. So this is virus 1, it's 10 kb long, and this is the small RNA profile along the genome. So in the ovary of this insect, there is a production of viral small interfering RNAs, which are probably downregulating the virus. So they are preventing that the virus replicates too much and kills the animal. On the other side, it does not clear up the virus. In fact, we can see that RPV1, together with all the other viruses that we found, can be transgenerationally transmitted to the progeny. So there is transovarial transmission. And here you can see that the viral RNAs in this animal are 22 nucleotide long. So it's the size range of the small interfering RNAs that are typical of the RNAi machine, of the RNAi pathway. So we are very excited about these, and we are now trying to prepare the manuscript and submit it. I want to show you 2 more examples of how the RNAi can be used in basic research to understand the function of genes in non-model organisms. Because as Valentino said, when we talk about the fruit fly, C. elegans, or mouse, these animal systems have been studied for 100 years. So there is a lot of tools that have already been developed. Now, the problem is with the other animals that are still necessary to understand thoroughly signaling mechanisms, signaling pathways, the role of genes and the mechanisms of gene regulation. So we need to study more model organisms, but we don't have the tools. So the RNAi was one of the first tools that allows really to pick up virtually any animal from the wild and knock down a gene. In this case, I'm showing you the case of planaria. This is a flatworm that is highly investigated. It's attracted a lot of interest because this animal can regenerate the entire body. So it's characterized by a whole body regeneration. If you cut it in 2, you will have 2 animals. So it's really wonderful for research. When you need more animals, you just chop up one of them, and after a few weeks, you will have more adult individuals. Now, what happens? If you do this, you cut and you let it grow, you will have 2 perfectly normal animals. But if you use the RNAi to shut down the wingless gene, for example, you can see that instead of growing a tail, now the planaria is growing another head. So this experiment tells us that the wingless is necessary to impose positional information along the anterior-posterior axis. It will basically tell the cells where they are in the body, and it will allow to place the head and the tail in the proper positions. Now I'm showing you another example that is from Elena Araujo group, and this is the work by Mateus. I think both of them are in the audience, so if you want to know more about it, they can tell you all the details. But this is just beautiful. If you can see that if you knock down the expression of this gene that they call Preto, you can switch the color of the Rhodnius from this wild type brown color to black. I find this really amazing. So again, this paper, they already put it in bio archives. So if you want to read it, it's already available online. Now, I've told you about the role of RNAi in model systems. How can it be used to cure human diseases or try to cure human diseases? The idea is very simple. Like there is some diseases like cancer where you know many oncogenes that are deregulated. In many cases, what happens is that a cell cycle regulator like P53 or others are expressed at higher levels, or they are constantly expressed, and that prevents the cells from stop dividing. This is one of the characteristics of tumor cells. So you can easily imagine that if you were able to downregulate the expression of p53 or any other oncogene by injecting siRNAs in the organism, like through the bloodstream or in any other way, these siRNAs could be uptaken by the cancer cell, and in theory should shut down the gene that is causing the tumor phenotype. Okay, so this is very easy to say. It's a bit more difficult to do because the siRNAs are not so stable in the bloodstream. I showed you that XRN2 nuclease. There is many other exonucleases in the body that can degrade the siRNAs, so they prevent these molecules from reaching the target cells and target gene. So there has been a lot of efforts in trying to stabilize the siRNAs. Many times they are included in nanoparticles together with lipids or sugar moieties. They are generally locked at the 5' and 3' ends. So basically, these are a lot of modifications that are introduced in the siRNA with a few goals. One is to stabilize the siRNA and protect it from exonucleases and enzymes that could degrade it in the bloodstream, but also favor the uptake from the cancer cells or from the target cell, and avoid that the siRNA is actually absorbed by cells that are not the target of the therapy. How can we do this? One example is given by the use of transferrin. So this is a protein that is used to coat the nanoparticles, and transferrin is recognized by a ligand that is expressed on the cancer cells. So what happens is that the cell will interact with a high affinity with the nanoparticle because it's coated with the transferrin. Now the endocytosis will bring the nanoparticle inside the cytoplasm, release the siRNA that can do its work and downregulate the target gene. Okay, so there is many studies that have been done over the past 15 years, 16 years, and they've brought to a number of clinical trials. I'm showing here only a subset, not only for cancer, but also for other human diseases. There is clinical trials ongoing. For example, for cardiovascular disease, there is a nice mouse model system for myocardial infarction. Maybe we can talk about it later on. Anyway, in all these cases, there is very promising results, but clearly the need is for more experiments, clearly, to make it safer and more effective. Now I will switch to the third part of the RNAi mechanism, and this is the most recent one. Again, it's centered on small non-coding RNAs. This time they are 18 to 13 nucleotide. And you can already see a difference from the other two branches. There is no dicer involved in their biogenesis. It's all done by argonaute proteins. Of course, there is, in all these pathways, there is a number of other cofactors that regulate the specificity. They help the loading of the complexes and so on and so forth. But let's say that these are the backbones of the three different branches. What happens is when piRNAs are produced, they are incorporated into a complex called PIRISC, and this is centered on an argonaute protein called PIWI. Now, this complex will recognize and degrade the target RNA, but also induce transcriptional silencing by modifying the chromatin. The best established function for this pathway is to silence transposable elements. Okay, I will go back there in just one slide. I just want to show you this study here. This was 2003. It's probably not the first time that the piRNAs were described. Actually, Alexei Aramin had already dissected the stellate locus in the fruit fly, which also relies on piRNAs. But this was the first study where small RNAs were sequenced on a large scale. And this coincided with the development of next generation sequencing technologies. Now, I told you that piRNAs are important for silencing transposons. Now, these are jumping genes. For those that are not familiar with this class of elements, transposons are regions of the genome, segments of the genome that can hop around. They can move from one side to another, either through a cut and paste mechanism or through a copy and paste mechanism. In both cases, so transposons were discovered by Barbara McClintock, that also received the Nobel Prize for this important discovery. Now, what is remarkable and is important about transposons, among other features, is that it is very important when they land. Like if they move around, it's okay, but depending on where they land, they can generate quite terrible situation. I will use again the example of Gregor Mendel. Valentino talked about the Mendelian laws. So you know that Mendel was using the P to study the segregation of traits. And one of the plants that he used produced a rough phenotype. So the seeds were wrinkled in certain combinations, while the wild type, let's say, this is the normal morphology. Now, this was the middle of the 19th century. And clearly Mendel had no idea what transposons were, not even genes, actually, it comes to that. But recently it was discovered that this phenotype is regulated by a gene called SBE1, which regulates the production of starch. So when the gene functions, you have round, nice shape. But if the SBE1 gene is impaired, you have the wrinkled phenotype because there is no production of starch. The gene doesn't work. This is actually what they discovered. There is a group that went to track back the variety of plants that most likely Mendel used by studying the trades in that period and what kind of peas were more commonly traded in the area where Mendel lived. And so they came up with this study where they showed that probably the wrinkled phenotype that Mendel was using was due to a transposon insertion into this gene. Now, why I'm talking about this story, aside from the fact that I think it's very fascinating, it illustrates a very important point. A transposon can insert into a gene, disrupt the function of the gene, and therefore cause a phenotype. And this is called insertional mutagenesis. Now, as far as we talk about peas and plants, it's not a problem, but when it happens in human, you can have a very pretty horrible situation. Actually, there is many diseases that are connected to the transposition or the insertion of transposable elements in important genes. For example, the disruption of BRCA DNA repair enzymes by allo elements is connected to breast and ovarian cancer, but we have many others. Here, for example, the Fukuyama congenital dystrophy, this is also caused by the insertion into a gene that is encoding for an enzyme that modifies dystrophin. That's a bit complicated, but in all these cases, the insertion of the element disrupts a gene that is important for human development, and this results, culminates in different types of diseases. The old situation is made worse if you think that roughly half of our genome is made up of transposon or transposon-related sequences. So going back to the RNA pathway, it seems that in animals, not in plants, this pathway is not active in plants, but in animals, this pathway evolved to silence the transposons, prevent them from mobilizing, and therefore protecting the genome. So the pathway guarantees the maintenance of genomic stability. I should add two more informations. Well, we can talk about the molecular mechanism and the way the PV induces heterochromatin formation later on. But it is also important to know that the transposition, the mobilization of transposons, does not only end up in insertional mutagenesis, but it can cause double-strand breaks, telomere fusions, and other problems that obviously affect the development of the organism. And this is particularly true in germinal tissues, which are dedicated to transfer the genetic information from the parents to the offspring. And regarding human diseases, I should also mention that another correlation that was found is that high expression of PV genes in humans are generally found in different types of cancer, and when these genes are expressed at high levels, the prognosis is very poor. So there's very poor outcome of the prognosis. The highest expression, the worst is the type of cancer. And I will close this chapter by also mentioning that piRNAs like microRNAs are being investigated as biomarkers. They offer actually advantages. We see just in later on how many different types of piRNAs you have in the human body. And so, same as for the microRNAs, in many diseases, many types of cancer, certain specific piRNAs appear to be either upregulated or downregulated. And so we can use, in theory, these characteristics to define the identity of a tumor, but also to follow its progression and also monitor the recovery after treatments. Now, I want to talk just quickly about long non-coding RNAs, only really just one or two examples, depending on if I have enough time, probably not. But so I will go back now to the 2000, and in the beginning of this century, there were some very important technological advances and discoveries that have boosted a lot scientific research. You will remember that clearly in 2000 there was the publication of the first draft of the human genome, but also in the same period, genome of other organisms like C. elegans, the fruit fly, and others were published and made available. Now, this first draft took a consortium of people, like there were many labs around the world that worked for more than 10 years to put together the first genome, the first human genome. But then a few years later, next generation sequencing were developed, which allowed multiple parallel sequence of DNA and RNA fragments. I'm not going to go into the detail of the chemistries and the protocols, but just to give you an idea, with these technologies, you can sequence a single person can sequence the human genome in a matter of days, and you can see here that the costs are relatively low. Might be a bit higher in this period of crisis, but anyway, so it's quite easy to sequence the genome. It's of course different question to assemble the genome, but sequencing, it's really not a big deal. And these technologies have been applied also to sequence transcriptomes, so we can now know, even at a single cell resolution, what types and how much RNA is produced. We can really see all the categories of RNAs that are expressed in each single cell. So what these discoveries revealed? First of all, that we have only, our genome is only for 2% made up of protein coding genes. The rest you can see is mostly transposon-related sequences, as this line element here, or intronic sequences. So a very small proportion of human genome really codes for protein. Yet, if we look at the transcriptome, you can see that 90% of the genome is transcribed. Now, this means that the vast majority of the transcripts expressed in the human cells and also in other organisms is basically non-coding RNAs. Now, you can imagine that for most cases, this might be just background noise. So it's just the result of pervasive or unspecific transcription processes. But these sequencing approaches also revealed a variety of non-coding RNAs that are really important in human development and often connected to diseases. I'm just showing you this slide to give you an idea of the variety. I told you that microRNAs are, well, they are more than 5,000. This is outdated, it's an old schematic. So there is more than 5,000 microRNAs in human, but if you look at the piRNAs here, you have 1 million different species. So this is why piRNAs are also a very promising class of molecules as biomarkers for cancer, to monitor cancer progression. So we have different types of non-coding RNAs. Now I will close by talking about only one of them. Maybe I will mention another one. Here is a list of those non-coding RNAs that have been characterized in more details. In many cases, what they share in common is that they act as scaffolds. So they are a scaffold to bring about proteins that will then do a certain job, which is, for example, modify the chromatin, induce heterochromatin, and other types of enzymatic activities. So we'll focus now on this exist gene RNA that is one of the most studied and also one of the most interesting one, because it's connected to the problem of dosage compensation. In human, as well as in many other species, we have a problem like that. Males and females differ in terms of sex chromosomes. You know that females have 2 X's and males are XY. Now, the Y, the small chromosome, mostly heterochromatic, not many genes. But the X chromosome here, this is actually a pretty important chromosome. There's many genes. And this means that in females, in theory, we should have 2 doses of those genes with respect to males. But what happens is that actually only one of them is active. And this story started in 1949 when Barr and Bertram observed what they called the Barr body in cat cells. So this is the nucleus of a female cat cell. And you can see this heterochromatic body here, which is not observed in male cells. Later on, Mary Lyon was suggested that this Barr body is actually inactivated X chromosome. Now, in the 90s again, like very important discoveries, it was found out that the X chromosome has a region, a locus called the XIC, X inactivation center, that produces a long non-coding RNA that is called exist. So what happens is that exist is produced from one of the 2 chromosomes, and it spreads in cis. So it will coat the chromosome, the X chromosome that is producing it. And it will bring about, will recruit many histone modifying and DNA modifying enzymes, which will then eventually heterochromatize the entire X chromosome and inactivate it. So in females, you will have 1 active X and 1 inactive. Now, this is a fluorescence in situ where you can see actually the 2 X chromosomes here in yellow, and only one of them is actually coded by the exist RNA. The other one instead is active. You can imagine that this will turn into the Barr body. One of the most typical phenotypes that is shown and is connected to the inactivation of the X chromosome is the color of the furs in the calico cats. So what happens is that these cats have these typical stripes like orange and black, or like 2 colors. And the reason why they display these patches is the fact that the gene that codes for the color is on the X chromosome, and there is 2 alleles. So one that will determine the yellow color, the other one that will determine the black or brown color. Early during development, one of the X chromosomes will be inactivated. And one important characteristic is that once in a cell, a certain X chromosome is inactivated, all the cells, the descendants, all the daughter cells and descendants of the daughter cells will have the same chromosome being inactivated. Maybe it's easier to imagine if you consider that one X is maternally provided and the other one is paternally provided. So if in a certain cell during development, the paternal X is inactivated, all the descending cells will have the paternal copy that will be silenced, will become the Barr body. And so because of this effect, while the cat is developing, you will have patches of yellow and brown because in some of the cells, the paternal copy is inactivated, and in the others, instead, you will have the maternal copy. Okay, so I'll just conclude with a couple of more slides. How much time I have? I don't have more time, so I have to skip this one. Just to conclude, I have told you that the sequencing of different genomes revealed that in human we have roughly 20,000 protein coding genes. And when we compare it to other more simple, let's say, organisms or animals, you can see that the number of protein coding genes is not that different. It's not dramatically different. So how come that the human brain, for example, is so complex? What is the origin of the complexity? And I'm not saying that the non-coding RNAs are the answer, but there is a very striking correlation when you compare the complexity of the organism, like a prokaryote that is down here, and a vertebrate, the human here is the first line here, the first bar here. So if you compare the protein coding genes, you don't see much of a difference along these groups. But if you look at the non-coding RNA content, there is clearly an increase in the content of non-coding RNAs, which correlates nicely with an increase in the complexity of the species. So maybe non-coding RNAs can explain these traits. And also, for sure, we have only uncovered the tip of this iceberg. There's probably many more non-coding RNAs that will be discovered in the next future, and whose function will be connected to important developmental events and also human diseases. Okay, I think I will stop here because I have already passed the, my time, and I'll be happy to take any questions.
Thank you so much, Attilio. It's a great talk. I will go and open the chat, but I have a question for you to start. So I'll just go ahead. So you've shown beautifully that there are different classes of non-coding RNAs. Now, do they cross-regulate each other?
Yeah, that's
in development, disease, physiology, and then how does it work for developing, you know, therapeutical strategies? Does it make it even more challenging, or what?
Yeah, so that's a really good question. Like, what is clear is that there is a crosstalk between different non-coding RNA pathways. And I had actually a slide that I was not able to show, but recently a new category of non-coding RNAs that are circular RNAs was found to be important to regulate microRNA levels. So these circular RNAs, they originate from back splicing. Basically, a gene is transcribed, and some of the transcripts, they fold back, actually they back splice, and they form circles. Now, in these circles, you have targets that can be recognized by microRNAs. Now, what happens is normally this microRNA will downregulate a certain gene, but since you have a circular RNA in cytoplasm, it will act as a decoy, as a sponge. So the microRNA binds the circular RNA. It doesn't do anything there, but it's removed, it's sequestered in the cytoplasm. So basically, you are relieving the silencing or the downregulation of the target gene. This is only one of the examples, but there is many crosstalks, not only between non-coding RNAs, but also between different epigenetic mechanisms. So non-coding RNAs will induce, for example, methylation of histone variants, or actually of histone tails. This will direct DNA methylation, but then the DNA methylation will recruit more non-coding RNAs. So there is generally self-reinforcing loops, feedback loops, which stabilize the function and potentiate the function. And this is important also for one additional attribute that is important in the development of the organism. That is the concept of robustness. Not only do you need to make a decision, like the microRNA or the non-coding RNA, not only has to make a decision, for example, whether a stem cell will differentiate or not, but the decision has to be maintained. This is being investigated a lot in embryonic stem cells. So once you make the decision that the cell has to start differentiate, you cannot go back. Otherwise, you will have defects, you will have a failure to develop the embryo. Now, going back to the second part of the question, that is, how does this impair or affect any development of clinical trials or drugs or treatments? Yeah, this is always a problem. In particular with microRNAs, the problem is that each microRNA has potentially hundreds of targets. And this is because the microRNA act through a very limited base pairing. You need only 4 or 6 nucleotides to match a sequence in the target gene, and that becomes potentially a target for the microRNA. And also you have the opposite situation, where the same gene is targeted by multiple microRNAs. So this is what is really complicated. And I remember a talk by David Bartel, who analyzed the function of many microRNAs, And in most cases, it turned out that they were responsible for very little differences in the expression levels of the target genes. And the question is like, why would evolution care about these changes? So just there is many open questions. The problem for the human disease is that many times there is correlations only between deregulated or altered expression levels for microRNAs and other non-coding RNAs and the actual disease. But there is no proof that that is the cause and not the effect. So this would be the first step, understanding what is the cause and what is just an effect.
Sure.
And then targeting microRNAs is not that easy. Like for miravirsen, you saw that there is a candidate drug, but there is not many others that are around. And even in that case, there were worries that it could affect other important processes. Again, that microRNA is important to protect the liver from hepatocellular carcinoma. So you cannot really mess around too much with these molecules. I don't know if I answered your question. Really,
I think you did, kind of. It's an open question, right? I mean,
yeah, some things
for sure. For sure. Let me
the
check
other thing
here.
Is I just had one more detail maybe that can help, and this is the same that is true for the CRISPR, the problem of the off-target effects. There was somebody that asked about off-target effect, and this is the same problem with the siRNA-based therapies. Like when you inject a small non-coding RNA in the bloodstream, your target, you know what is your target, but it is known that RNAi has off-target effect. So you could be knocking down other genes that are important for development. And this is also the worry related to the two twins that have been engineered in China. And the worry is that the CRISPR that was used to knock out the receptor of the HIV, this was the aim. The CRISPR not only knocked down that gene, but also others that might be important and might have consequences.
All right. Manuel, do you want to go ahead and ask your questions? Your microphone is off.
Sorry. Okay, first, thank the presenters again, and thank you, Katarina, for the help. And quick question, just I may be crazy, but you did mention, Attilio, that there are several strategies to try to send double-stranded RNA or interference RNA into targets for therapeutic reasons. Would it be possible to use CRISPR to make artificial gene and to insert a gene using CRISPR, using an interference RNA gene using CRISPR, like a synthetic microRNA gene in cell?
Yeah,
and the second question, it's just more than a question, or let's say a provocation for you. Which of the strategies would be helpful in COVID, in the fight against COVID? You mentioned that some virus have a defense mechanism based in the interference RNA. So just two questions.
Yeah, these are great questions. Maybe the first one, even Valentino can help me with. There is strategies that are based on the CRISPR, which allow homologous recombination. So you can introduce gene cassettes in the cell. And so if I understand your idea will be to introduce something in the genome that would actually act as an RNAi, programmed RNAi mechanism. So this is in theory possible. Actually, it will be even doable if you think that they have already engineered some embryos. It will be ethically criticized, of course. And I think that using the homologous recombination, from what I've heard, it's a lot more difficult than inducing mutation. So if you want to edit a gene using CRISPR, it has a certain degree of difficulty, but introducing by homologous recombination a cassette seems to be more complicated. But again, this part Valentino knows certainly a lot more about it. And as for the COVID, yes, so the organism, there is insects, they have an RNAi mechanism to reduce the viral load. They don't remove the virus. We see that also in rodents. The viruses have been there for 10 years. We have evidence that they've been there for 10 years. So the virus is not removed. What the RNA does, it reduces the viral load so that the organism doesn't die. So it causes what are called persistent infections.
Okay.
The ones that I showed you are RNA viruses, and it's the same for Zika, dengue, chikungunya. These are all RNA viruses. Now, the coronavirus is a lot more complicated virus. It's a bigger virus. I think it's 30 kb. And actually, a month or two ago, a friend of mine from the Pasteur Institute gave a talk, and at the very end, he's actually working on C. elegans, but he has worked on the coronavirus to check for the presence of small RNAs. So he didn't find some small RNAs that target these actually that are produced by the virus. But again, this is only in the beginning, and it is to be determined what is the function of these molecules. Mm-hmm. In theory, you could think of introducing siRNAs against the COVID in the people. The problem is that in adults, we don't have an active RNA interference. This is because the double-strand RNA causes the activation of an interferon response in adult cells, which ends up killing the cell. So paradoxically, you have RNAi early during development in the embryo because it's less destructive. So you can remove a virus in the embryonic stage, and you don't lose, because you don't want to lose stem cells. But then as differentiation proceeds and the organism is growing, the RNA is replaced by other mechanisms like the interferon and other immune responses. So bottom line, in the adults, this will not really work, and it will be difficult because it's sort of easy to target the siRNAs to the liver, but reaching other organs and tissues is a lot more complicated. So this is also one other challenge for the future. I don't know if Valentino wants to add up something on the.
Yeah, I mean, you already pretty much answered. Like, it's with using CRISPR, it's very easy to cut and make a small mutation, but when you want to insert something with HDR, it's way less efficient. So in those applications that are trying to use CRISPR to add something, it's usually done ex vivo in therapy, where you just take out cells, you modify, you select the ones that have the correct editing, which can vary between like 3 to 10%. And then once you re-amplify those, you put them back. But that's, you know, it's just one tissue, and usually it's for a specific function. So I don't know whether modifying a small amount of cells could have an impact on the whole body. It's possible. Maybe in the future, but with the current technology, probably not.
One thing that can be helpful in this case, and really just engineering only some tissues or some cells might work, is that many of these non-coding RNAs can be released in the bloodstream, and they are stable. Like microRNAs are very stable in body fluids, in urine, blood, plasma, so saliva. So that's also what makes them very promising candidates as biomarkers. And so in case of, you know, the idea that Manuel had, if you manage to express small RNAs against the gene or against the virus from a certain group of cells, and these are released in the bloodstream, then they might work. The problem is that it's a lot more complicated than it sounds like.
I don't seem to see more questions in the chat. There's Anna Celia that asks Attilio to provide the PDF, because the tables were with such a small letters that she wants to read them all, and that would be extremely helpful if you could share the PDF. And again, I would really like to thank both speakers, both Valentino and Attilio, for this wonderful session on CRISPR and on coding RNAs. I certainly learned a lot, and I thank you so much for your precious time, and everybody that was present here today. We will come back for new sessions in a 2-week time, right, Manuel?
Thank you so much for an invitation.
Thank you.
Thank you, Catarina. Thank you, Manuel. And
Okay. Okay. Good night, everybody. It's night in here.
Ciao, Valentino.
Bye. Bye, everyone.
Bye. Thank you.